I'm John Gribben from Barts in London, and this is iwNHL 2018 in very sunny Nice in southern France. We've just had a very interesting session on immunotherapy, a huge area, and focusing particularly on checkpoint inhibition. It's true to say that as hemato-oncologists, we don't like getting beaten by solid tumour guys, but it seems like we've got behind where they are. And with the notable exception of Hodgkin's disease, I'll come back and talk about in a moment. Robert, we do seem to be not getting the kind of successes that we were hoping and expecting to see with checkpoint inhibition. You were looking at PD-L1 going beyond and thinking about combinations. Is that what it's going to take in non-Hodgkin's lymphomas to look at combination therapy? And why do you think that checkpoint inhibition by itself in most cases in non-Hodgkin's lymphoma is rather disappointing?
Well, one simplistic view of looking at it is that, with the exception of Hodgkin lymphoma, PD-L1 or 2 is not overwhelmingly expressed in non-Hodgkin lymphoma, or maybe also with the exception of primary mesenchymal large cell lymphoma. And it's in these tumor types with a higher PD-L1 or 2 expression, you tend to see stronger efficacy with the PD-1 inhibitors. So maybe in the other non-Hodgkin lymphoma, you do have to go with combination routes to try to amp up the immune signal. The other thought is that, in a lot of non-Hodgkin lymphoma, especially the aggressive B-cell lymphoma, the kinetic of disease is very fast. Whereas in solid tumor, it may not move as fast as we do. And sometimes I do wonder with immunotherapy, maybe you need to add something, perhaps chemo or other things, to slow things down a little bit to give it time for immunotherapy to work.
Do you think much of impact of what we've previously treated the patients with has any impact on the T cells which are there, or do you think that would just be the same in the solid tumor setting and that they've probably also damaged the T cells by the chemotherapy that's previously been given?
I think it's hard to say at this point. I think we had contradictory evidence at this point. As Steve was saying, in the nivolumab study, when you treat it in multiple relapse patients, the efficacy is about the same as you give it upfront in a window study. However, in the study that we're doing, when we give it as a second-line therapy, we seem to see a stronger efficacy. So I think the jury's still out on that.
Now, Steve, the one noticeable exception, of course, is the studies you've led on nivolumab and pembrolizumab now also in Hodgkin's lymphoma, where for reasons that we thought we understood, it was very logical this was going to be a cancer that was going to work very well with immunotherapy, but still some unanswered questions of why it works just quite as well as it does. You also alluded to in your talk the issue that lots of these patients are responding and then subsequently relapsing, suggesting that either we go back and retreat again or that we have to add in other things. So what are your thoughts about where do we go beyond nivolumab in Hodgkin's?
Yeah, I think as you point out, the exciting thing is there's been really nice results with PD-1 blockade, but I agree with you. I think our challenge is just what to do next that would really further augment that benefit. And I think there are a few options. I think option number one is to do something that would actually further activate the immune system. So you've taken away the inhibitory signal, you're relying on the endogenous immune system to do its thing. Could you give the immune system a little push? So there are other kind of activating agonists that you could consider. I think the other thing that we're finding is it's really important still to go after the tumor cell because the malignant cell really defines the environment. So if you don't actually change that, the problem is the fact that even though the immune system and the immune cells may be activated, they're still running into the same barriers that are preventing them from doing a good job in the first place. So I think changing the environment and then activating the immune system on top of inhibiting the negative signals may be a configuration that we can really work on.
So you went over a whole variety of different agents which are in early phase development within the setting. Which of those kind of is kind of exciting you most in terms of there's an opportunity to build on this? Anything in particular?
So I think there are a lot of exciting combinations, but the point you made earlier is important in that even though the solid tumor folks have led in some degree, not all of the lessons that they've learned are 100% applicable. So to give an example, combinations of PD-1 blockade and CTLA-4 blockade have been very effective in melanoma, for example. Not so true in hematological malignancies, and part of it might be that now the immune system is the malignancy, and I think the T cells and B cells naturally talk to each other, and that may be different to, let's say, solid tumors where the immune system and the tumor are different. So I think as we're kind of looking to how to do this, I think there are combinations that are promising, but it won't be across the board. But I do think the combinations that are potentially promising are going to be the ones where we combine different ways of modulating the immune system. But we're a little early to actually know which ones are proving to be promising, because those studies are just starting now.
Now, none of us kind of addressed this in our talk specifically, but it came up in the discussion afterwards. This question you already raised there about T-cell, B-cell interactions, and remembering, of course, there's the potential for T-cell help as well as, T-cell activation against the tumor. How do you think we need to be thinking about how we get the sorts of T-cell activation we want and don't land up leading to enhancement of tumor cell growth?
That's a fabulous question. I think that's a very challenging one, because I don't think we completely understand what are the messages that are beneficial to the tumor cell and what are the messages or activities of a T cell that inhibit the malignant cell. I think we have some understanding of that, but exactly how to modulate that is difficult to know. I think one of the things Rob shared about today, which I think is interesting, is if you actually use a targeted therapy that kills the malignant cell directly. So he was talking about brentuximab vedotin, which actually targets the Reed-Sternberg cell at the same time as modulating the immune system and the efficacy thereof. I think that's a promising approach where you're getting the malignant B cell suppressed at the same time as activating the immune.
Yeah, I was actually thinking about that when we were being asked those questions, that maybe there's even the potential that stimulating the B cell might make it more susceptible to a chemotherapeutic approach killing. So there might be an even better rationale for thinking about the two together in combination. And of course it makes sense to look for a targeted killing of the tumor cell, because you don't want the chemotherapy to be having an effect on the other cells within the microenvironment.
Yeah, and so certainly, just like how brentuximab vedotin is targeting CD30 in Hodgkin lymphoma, there's a variety of antibody conjugate that are emerging for non-Hodgkin lymphoma as well. And I'm not aware of any trials that's combining them with, immune inhibitors, checkpoint inhibitors, and perhaps it's time to evaluate those options.
So there we have it. What we know from this session is that there's a lot of excitement in the immunotherapy field, a lot more questions, and what I'm suggesting is we're going to have another session on immunotherapy next year at iwNHL 19, where we're going to be coming back and hopefully a little bit further forward than we were today in many of the questions that we had. So thank you very much.